damos paso al proyecto Artemisina. Las 3 charlas que vienen a continuación nos mostrarán cómo diversas instituciones se unen para aprovechar los desarrollos de la biotecnología y así mejorar el acceso a los antimaláricos. Damos la bienvenida al Dr. Patrick Westfall, científico de Amherst, quien ha estado involucrado en el proyecto de Artemisina desde sus comienzos. Estudió genética en la Universidad de Purdue y posteriormente realizó un doctorado en botánica en la Universidad de Georgia. Antes de vincularse a Amherst en 2006, formó parte de grupos de investigación en biología molecular, biología celular y bioquímica en las universidades de California en Berkeley y San Francisco. Damos la bienvenida al Dr. Patrick Westphal.
Good morning. My Spanish is even less than Dr. Stewart's, and I apologize for that. But I am going to do my best to talk slowly, enunciate, and tell you a really fun story that we've developed through a collaboration with the University of California, Berkeley, Amherst Incorporated, and the Institute for One World Health. And what we've done to use synthetic biology to produce a precursor to an antimalarial drug called artemisinin. Before I get started into the molecular biology, I just kind of want to talk a little bit about Amherst. Amherst was started in, like I said, as a collaboration between University of California, Berkeley, located here. This is our headquarters in Emeryville, California, but we're also all over the world. We have a pilot plant and facility down in Campinas, Brazil, and we're doing global manufacturing to produce actually a biofuel, which is located everywhere from the central U.S., Lyon, Spain, and also down in Brazil. These are the founders of Amherst. In the tan jacket here is Jay Kiesling. He's a professor at University of California, Berkeley, who started the project. And the gentleman in the background are the 3 founders. Jack Newman is our chief science officer. Neil Renninger is our chief technology officer. And Kincaid Riley is one of our consultants. These are the guys who were in Jay's lab as postdoctoral researchers who had the idea to start Amherst and who were participating, wrote the grant to the Bill and Melinda Gates Foundation to get Amherst off the ground. We talked about this kind of central dogma, this reductionist idea of how you do biology, and I have a slide that says a very similar thing. How it was kind of done before is you'd identify a single gene from some organism. You engineer this gene into some sort of microbial host, in this case E. coli, and you make a drug, in this case insulin, to treat diabetes. This is kind of how things were done before. Synthetic biology is kind of like this on steroids. And the idea is that we look at all the central metabolism and we identify pathways or nodes within central metabolism, isolate all the genes, all the proteins, engineer this into some sort of host. In our case at Amherst, we use Saccharomyces cerevisiae. It's the same kind of organism that you can use to make beer and bread. It's a great fermentation organism. And we've developed a platform that can make all kinds of things. A lot of Amherst's technology right now is focused upon making biodiesel. We also have a program focused on making jet fuel. We can make basic molecules that can be used for a variety of purposes. In this case down here, an example of making renewable squalene, a compound that normally comes from shark liver. But the one that I'm going to talk about today is artemisinin. It's an antimalarial drug. And I'm going to tell you the story of how we developed this platform technology to make a precursor to this drug and how we're making it available through partnerships with One World Health and Zagaya. A little bit about malaria, although we've already had a great introduction. It's carried the, make sure I go the right direction here. Malaria is transmitted by female Anopheles mosquitoes. Here's a plasmodium parasite that causes the disease, and in this case, infected red blood cells. There's a little bit of background, and again, these numbers might be somewhat controversial, but malaria does affect about 109 countries worldwide. In 2010, when the World Health Organization was able to get a count, there was estimated to be about 216 million cases of malaria, about 665,000 deaths. Most of these deaths occur in children under the age of 5. And they estimate on average that one child dies of malaria every minute in Africa. The drug I'm going to talk about, artemisinin, is actually not a new drug. It's been around for a very long time. Back in 168 BC, the first mention of artemisinin, and I believe this is the symbol for it down here, was mentioned as a treatment for hemorrhoids. A little bit later, in 340 AD, it was also found again as a prescription for fevers, most likely malaria. It didn't really come back on the radar until 1967, when China, who were supporting the Vietnamese against the Americans, started a top-secret project called Project 523. They wanted to actually go into a plant that they used through traditional kind of Chinese medicine. They would brew a tea of this plant to treat their soldiers who had malaria, and they wanted to find the active ingredient. The active ingredient, which is known as artemisinin, was isolated in 1972, but really not a whole lot else was done by it. The current malaria drugs, the quinone drugs, were highly utilized and well known, but as we all know, the parasites starting to become resistant to the quinones. In 2004, the World Health Organization actually recognized artemisinin in combination with other therapies as the frontline treatment for treating cases of uncomplicated malaria. If you wanted to actually treat malaria, you need somewhere between 200 and 300 million artemisinal combination therapies treatments per year. It would require about 150 to 200 tons of artemisinin produced. And up until this year, the only source of that production was from plant material. Artemisinin is produced in the trichome parts of plants, and you have to harvest the trichomes and extract the artemisinin from that. If you wanted to treat all these cases, what you have to see is about a 2 to 3x increase in production, a decrease in stabilization of the price. The price of artemisinin highly fluctuates. And really, one of the main goals of this project was to eliminate stockouts. It's very, very sad that you have a mother whose child has malaria, walk into a drugstore, has the money to pay for the treatment, and it's just not available. It's not the right season or right time of year. So, again, one of the main driving goals of the artemisinin project is to eliminate this fact. So when anybody goes into a drugstore with malaria, they can actually get the drug. It'll be available. How we're approaching this from the fermentation, from the molecular biology side, it's nice to draw a comparison to kind of the traditional cultivation of Artemisia annua, which is the plant that produces artemisinin. This is a field in China growing Artemisia annua. It's got about a 10 to 12 month growing cycle. It's highly variable depending on weather, rain, wind conditions, things like that. They extract artemisinin from the plant. This is an example of the powdered artemisinin. And then they derive it. So this structure, sorry about that. This structure right here is artemisinin, but then they can actually make more effective forms by a chemical derivation into more effective treatments. The Amyris approach actually involves engineering microbes, again, Saccharomyces cerevisiae, but then through a fermentation cycle, actually producing this in fermenters. And the main difference where we can really have an impact is this 10 to 12 month growing cycle is highly variable. This is a 2-week fermentation cycle. You can just do it in 2 weeks, produce the raw material, which in this case is called artemisinic acid, and then through some simple chemical steps actually make artemisinin. So we can have a constant supply of production to provide for the shortfall that comes from getting it from the plants. All right, I'm going to switch gears and really get into the molecular biology. I'm going to use a lot of different terms and kind of go back and forth, so I decided to provide this kind of primer that will at least identify the compounds, and as much as possible try to color code things so you can see as I go through what part of I'm talking about. Artemisinin, we isolated the plant genes, or actually Jay Kiesing's lab isolated the plant genes needed to make artemisinin. There's several involved. The first metabolic intermediate, though, that is not naturally produced in yeast is called amorphidine, and it's this compound shown in red here. For the purposes of this talk, think of amorphidine as kind of the precursor. It's non-toxic to the cells. It actually is a really good indicator of how much carbon is flowing through our system and how much product we can make. It's a proxy for actually making the final products. Amorphidine is converted into artemisinic alcohol through a series of enzymes called P450s and reductases. It's an oxidation-reduction reaction involving the transfer of electrons. Artemisinic alcohol then is converted into artemisinic aldehyde, again by a P450 and a CPR. And then there's yet even a third oxidation-reduction reaction to make artemisinic acid. Now this is the final product that Amherst is interested in making. This was very easy to produce because we understood the steps along the process. It makes a great raw material, but it's not the final material. Through chemistry, we actually get artemisinin, which is the compound shown here at the bottom. And it's thought that it's this peroxide bridge in the middle of the seven-member ring that's effective against the parasite. But for our purposes, this happens kind of beyond the biology. The biology is focused on these pathways with these intermediates with the idea that we're going to make artemisinic acid. Go back to the idea of synthetic biology and isolating whole pathways. The pathways that I'm going to talk about today out of this central metabolism is called the mevalonate pathway. In yeast, it's normally used to make ergosterol, which is a steroid that affects membrane fluidity. In humans, the equivalent would be ergosterol. It consists of 8 enzymes, and we kind of break it into different parts of the pathway. The top part of the pathway starts with a metabolic intermediate called acetyl-CoA. It goes through a series of steps to make mevalonate, which is why we call it the mevalonate pathway. From there, it undergoes a series of phosphorylations and reactions to make a precursor molecule called FPP. And this is really the node that we focused on because normally this goes on to make ergosterol, again, what the yeast uses. This is also the stepping off point. This is what the amorphidine synthase uses as a raw metabolic intermediate to produce amorphidine and start the process on the way to making artemisinic acid. I've taken that one part of metabolism, kind of blown it up here, and I want to talk about some of the basic work that actually occurred at UC Berkeley and Jay Kiesling's lab to get the project off the ground and started. So again, you have the top part of the mevalonate pathway here making this intermediate. The bottom part of the pathway is going to make FPP, which again normally goes on to make ergosterol in yeast. And some of the first molecular biology engineering that the Kiesling lab did was it actually reduced the amount of ergosterol made by downgrading a gene called ERG9 and replacing that ERG9 gene with the amorphidine synthase, so we can actually look at the amount of amorphidine we could produce. The other thing we had to do in this pathway is get rid of regulatory mechanisms that existed within these proteins, and we did that for a couple of known bottlenecks. The first one here is this reductase step where we replaced this HMGR CoA reductase with 2 unregulated versions. They were truncated. They got rid of their regulatory region. And we put 2 copies of this particular gene in the pathway such that we relieved the bottleneck and we could get more flux going from acetyl-CoA to this mevalonate intermediates. The next step that the Kiesling lab did was take the final kind of terminal step in the pathway, which is also limiting around this gene right here, ERG20, to increase the amount of FPP that could build up in the cell. The rest of the pathway was regulated by introducing a kind of ubiquitous transcription factor, a dominant mutation of this transcription factor called UPC2-1. And this transcription factor actually upregulated these genes here, ERG13, ERG12, and ERG8. But that wasn't under our control. This was just kind of a general fix. They did this, and they published a paper back in 2006 in Nature Biotechnology, and what you can see is you take a strain that's just expressing amorphidine synthase that barely makes any detectable amorphidine, that first committed step. By making these genetic changes, they were able to actually achieve about 150 mg per liter of amorphidine, which is pretty good. But what we're really interested in, again, is this is a nice first step. We want to get to this final step here by going through this oxidative reduction steps using these enzymes called P450s, represented here by the CYP71AV1. And these are actually out of the Artemisia annua plant. And then it's reductase to help it go through this 3-step electron transfer reaction. We engineered this into yeast. We put the P450 and the CPR in, and we got what we wanted to see, which was very little amorphidine made, but the majority of the product was actually artemisinic acid we were after. But to put this in context, if you really want to make an impact by making artemisinic acid that can be converted into artemisinin, we were nowhere close to our target, which was around 25 grams per liter. We were more like 2.5 grams per liter. So we had to have a 10-fold improvement in production to really hit considerable economics to make this available. So we went back, we looked at the pathway and said, okay, there's certain things in the pathway that aren't upregulated. We have known bottlenecks which we can control, we can turn up, we can express a lot of. But other parts of the pathway either were not under our control, they were just the native genes, or under the control of this kind of ubiquitous transcription factor. So we said this is synthetic biology, we can engineer the entire pathway, not just one or two genes. So let's do that. And to do that, the first thing we did is got rid of this transcription factor and really made what we kind of refer to as super yeast, where we took the entire pathway and put every single gene under our control. They're all inducible. We add a small molecule. It turns these pathway genes on up 1,000-fold. In addition, we actually added the biggest bottleneck in the pathway we'd identified was this HMG-CoA reductase. We put a third version of this unregulated gene in the pathway to really, really crank it up. We wanted to, again, kind of assess how much carbon's flowing through the pathway. Are we even getting close to the amounts of product we want to make? To do this, we just looked at this very first molecule. Again, this amorphinine is non-toxic to the cell. It's a great proxy for how much carbon is flowing through here to see if we can actually hit our target goals. And lo and behold, not only when we looked at amorphinine were we able to hit the 25 gram per liter goal, we actually shot right by it and made up to 42 grams per liter. So this showed that this modified pathway was actually capable of making up to 40 grams a liter of product, which would definitely make the process economical. It's those last three steps, though, that were the problem, because when you compare the original strain of amorphinine producer, what we call yeast 1.0, we can make about 400 mg per liter in a shake flask. This is our new strain. This is, again, in shake flask, not fermentation. It can make pretty close to 2 grams per liter, so about a 5-fold increase in amorphinine. But you add those last three steps in, that P450 and CPR oxidation reduction step, there wasn't much artemisinic acid made. We had a backup plan. The chemists down in the lab who were just as committed to this project as we were said, hey, look, we can make lots of amorphinine. Maybe there's a backup plan through chemistry. And they actually came up with a process that was fairly efficient. But the problem with chemistry is each one of these steps costs extra money. And to be economical, it would be really preferable for the biology to take care of these steps, at least down to artemisinic acid. So we sat down as a group, and we just kind of hypothesized, okay, what are the main things that could be going on here? What's wrong with the system? Maybe just expression of these proteins. Maybe the P450 and the CPR being expressed are gumming up the works, are messing up the Saccharomyces strain. It didn't make a lot of sense. Saccharomyces has its own set of P450s. They're expressed. Everything was localized right, but it was a possibility. Another very obvious possibility was toxicity. The actual product we were making, artemisinic acid, was toxic to the cells. A third possibility was improper oxidation. Remember, there's intermediate steps in there. If we're not efficient in our conversion of amorphinine to artemisinic acid, those intermediates could be toxic to the cells. Oxidative stress. P450s, because this electron transfer, if they're not coupled right, if that CPR molecule is not coupled right to that P450 protein, they'll throw off a lot of hydrogen peroxide and cause oxidative stress, which is very damaging to the cell. It could have to do with redox balance. There is a step where you have NADPH and NADP as part of the coupling reaction, and if this couple, again, if the P450 and the CPR don't fit together quite right, you can throw off the redox balance of the cells. And then the last possibility was maybe we're actually missing some enzymes in the pathway, and there's an enzyme called cytochrome B5 that in the literature in some cases has been shown to help couple this P450 and CPR together more efficiently, as kind of acting as a bridge. And what it allows you to do is if the cytochrome B5 is present, you can actually change the ratio of P450 and CPR. In nature, usually there's about 40 P450s to 1 CPR that occur in nature, and at the time we were expressing these at approximately equal levels. So the first step was to actually go out and identify the cytochrome B5 to see if it could help couple the P450 and CPR together. And what you can see here is in the case of when the P450 and CPR were expressed at approximately equal levels, we can make artemisinic acid, but when you actually dropped it to what would be more like native levels, we took a hit in artemisinic acid production. By putting that cytochrome B5 bridging protein enzyme back in there, we were actually able to get a significant bump in production and actually lower the overall toxicity of the cell. We actually saw by going from this step down here, we reduced the oxidative stress burden, and the oxidative stress burden didn't jump up here, but artemisinic acid production did. Okay, so we're back to our model. Everything's overexpressed, but now we've modified this part down here where the P450 is still highly expressed. But we've reduced the CPR, and we've added the cytochrome B5, which will bridge those two proteins. However, again, in the grand context of things, there's still this huge delta. Here's how much amorphidine we can make in a shake flask. We're not even close with artemisinic acid. So, what's going on here? This is another visualization of that, and Kirsten Benjamin, one of the scientists in the project, said, hey, look, we're measuring artemisinic acid, we're measuring very little amorphidine, but what about the intermediates? If the P450 isn't performing all three reactions very efficiently, but if it's only performing one of the reactions very efficiently, we should start seeing intermediates build up, and that's exactly what we saw. So again, this is our model. This P450 performs these three reactions. If either artemisinic alcohol or artemisinic aldehyde is building up in the system, that tells us that this P450 is not functioning fast enough to actually keep up with all that carbon flowing through the system. In particular, we were worried about artemisinic aldehyde because even more so than anything else in the pathway here, this is highly, highly toxic to the cells. So Kirsten and her group went back and actually measured that, and sure enough, a lot of artemisinic aldehyde is building up in the cells. In fact, when we looked at viability, only about 20% of our entire population in the fermentation tank was actually alive. We think a lot of that was due to this buildup of artemisinic aldehyde. Now, luckily, and this is really important, we were talking about collaborations and outreach. We have a lot of infrastructure in Amherst. We had a lot of really great scientists, but we kind of committed to our own ideas, and we decided to go out and do a little bit of outreach, and a little bit of outreach came to us. This is Pat Covello. He's a professor up at the Canadian National Resource Center in Saskatoon, Canada, and he had continued to do work on the plant, Artemisia annua. And what he had discovered was that although that P450 is very good at converting amorphidine to artemisinic alcohol, that very first step, there was actually other enzymes in the plant that could perform the last two steps much more efficiently. And he called these artemisinic aldehyde dehydrogenase, or artemisinic alcohol dehydrogenase, and artemisinic aldehyde dehydrogenase. We went back to our model and said, okay, if these are the steps that are stuck, what if we replace these last two inefficient steps with Dr. Covello's new genes? The alcohol dehydrogenase and the aldehyde dehydrogenase. Will that make a difference in our pathway? And it really, really did. So again, these are kind of shake flask productions. They show all the intermediate buildups. Again, switching from the high CPR P450 ratio to a low one, adding cytochrome B5. But the minute you started putting those extra genes in from Dr. Covello's lab, you see we got rid of the artemisinic aldehyde almost immediately. And again, that was the most highly toxic part of the cells. And our cell viability jumped up to 80% of the cells being alive. And you add both genes in, and you have very little of the alcohol building up too. Back to the model. Again, we reduced the CPR. We have the bridging enzyme cytochrome B5. We replaced these last two steps in the pathway with the alcohol and aldehyde dehydrogenase. And now we have a much more efficient pathway. When you put that in fermentation, all of a sudden we're where we need to be. Through process controls, through this genetic engineering, we were able to actually hit the 25 gram per liter mark and hit what we think will be sustainable economics for production of artemisinic acid. So to summarize some of the molecular biology I talked about, one of the big breakthroughs was this idea of engineering the entire mevalonate platform, overexpressing every gene in the pathway, putting it under our control. We can turn it on, we can turn it off when we want to. And this allowed us to actually exceed the target goals for amorphidine and eventually artemisinic acid. The second thing we learned was that this high level of P450 and CPR caused a lot of oxidative stress. We had to get that ratio just right, identify that bridging enzyme to make sure that the cells weren't just dying because of the overexpressed enzymes. And finally, and this is really through this collaboration with this Canadian lab, even though this P450 and CPR could do all three steps, in nature it didn't actually perform it, only performed one of those steps. And it was through this work of Dr. Covello we were able to identify these additional enzymes that allow us to get rid of those toxic intermediates and make very significant quantities of artemisinic acid. And so where does this leave us? This is an article from 2010 in Nature talking about malaria and the demand for the malaria drugs that's really soaring. Malaria is on a comeback. These known drugs are not as effective anymore. Artemisinin is really the only drug out there that's novel, and we want to make sure that it's available to people who need it. This was a Boston Consulting Group survey that they performed for us back in 2009. And what you can see is that the impact of the artemisinin project that Amherst is working on really is going to be seen in 2012 and beyond, really starting this year. And that's this idea that these new technologies are coming online and will provide an additional source of artemisinin. And then finally, to, set 2A up, this is a press release from the Institute of One World Health, and I just kind of want to highlight the bottom line here, which is this idea that the work that I talked about today is the intent is to commercialize these ACTs containing the semi-synthetic artemisinin this year. And again, 2A and K will talk about that a little bit, hopefully. To finish up, obviously a lot of people were involved in this work. I want to thank people in Jay Kiesling's lab. Eric Paradise was the first person to start engineering these pathway genes. Dae-Kyun Roh actually identified many of these genes from the plant, from the Artemisia annua plant, and optimized them for expression in yeast. The Institute for One World Health, who helped oversee the grant from the Bill and Melinda Gates Foundation. Kim and Rowe from Zagaya, which is a nonprofit spinoff of Amherst. And then on the Amherst side of things, I particularly want to point out Chris Padden, who is the program leader for this project, kind of rallied all the troops, kept us all on track, and Kirsten Benjamin, who was the other technical lead and the scientist, who was really key in identifying those toxic intermediates and really allowed us to make the breakthrough progress that we did. So with that, just a picture of where I live. This is Amherst right here in Embryville, California, right across from San Francisco, where One World Health is. This is the building. Amherst is on the bottom 2 floors of this building, and actually Jay Kiesling's lab is on the top floor of this building.